Alright guys, let's go ahead and push on through with Chapter 6. I don't know about you guys, but I already feel more comfortable with this chapter. I'm more familiar with this information. It seems more familiar to me. I think I've studied this perhaps a bit more, so hopefully you feel the same way. Hopefully you feel like this is a bit of a reprieve after Chapters 4 and 5. I know that beat some of you up just a little bit with the biochemistry. So this is information that you probably started with in probably grade 7, and then in perhaps grade 9 or 10 when you had biology again. We're just going to go perhaps a little bit deeper than you did the first time, but it shouldn't be brand new information. So already we've looked at some of the cell parts. We started with the cell membrane, and then we went straight to the nucleus, which is the head or the main control center. And then we talked a little bit about some of the ribosomes as well. And now we're going to go into the bags inside of bags inside of bags. So the next portion is Concept 6.4, and it says the endomembrane system regulates protein traffic and performs metabolic functions in the cell. And when they say endomembrane system, they basically mean the bags within bags within bags that we were talking about. And the parts of the endomembrane system that we're going to look at include the nuclear envelope, which is like the membrane that surrounds the nucleus itself. And then we have a couple of the organelles, the endoplasmic reticulum, the Golgi apparatus, the lysosomes, vacuoles, and the plasma membrane. And these components are either continuous, meaning they're connected together, or they are connected via what we call vesicles. And a vesicle is just where a part of one of these sacs or membranes will pinch off and then float to another sac or membrane and then combine with that one. So again, we're still talking about just bags upon bags. So a vesicle is simply a pinched off portion of one of these membranes that is used as a transportation kind of device, kind of like a transportation pod to get material from one place to another within the cell. We're going to start with what's called the ER, or the endoplasmic reticulum. It's basically a factory or slash highway system of the cell. Oftentimes it's thought of kind of as a highway system because it's a huge membrane and it contains lots of infolds, and it takes up a fair portion of the cell. The endoplasmic reticulum is more than half of the total membrane material or area within the cell, and it is continuous with the nuclear envelope. So where the nucleus, the nuclear membrane, begins or separates the nucleus from the cell, that's where the ER connects. So the ER is connected to the nucleus. It is continuous with the nucleus. And there's two specific regions we're going to look at on the ER. Parts of the ER are called smooth, and that's because they don't have the little ribosomes embedded in them. And parts of the ER are called rough, and that's simply because they have a surface that looks rough or dotted because they have ribosomes embedded in them. Here's a cool picture. This down below, of course, at the top is just a model or a drawing, and then down below is an actual microscope picture. So you see the nuclear envelope. They have the cutaway here. This would be like the outer portion of the nucleus. You can see the pores there, the openings, and you can see where the ER connects to the nuclear envelope and then continues on out into the cell from there. The smooth portions of the ER up here have no ribosomes. The rough ER, which is the majority of it here, is studded with the ribosomes. And here you have transport vesicle, is just a little bubble that has pinched off and is being used to move material from one place to another. The smooth ER and the rough ER do have some similar functions, although we'll kind of separate them out. The smooth ER is used for synthesizing or creating lipids. Remember, lipids are things like oils, the phospholipids that make up the cell membrane, steroids, so some of your hormones, like the various sex hormones in humans, which are types of steroids, those are created in the smooth ER in portions of the body. They metabolize or break down carbohydrates. They also have a job to do with poisons or other materials that the body needs to change or get rid of. So, for example, if you were to drink alcohol in and of itself is somewhat toxic to the body. So what happens inside the liver is that you have the cells of the liver, their smooth ER actually changes the alcohol to make it so that it's more water soluble so that it can be flushed out of the body. And they do this by adding a hydroxyl group onto the end of that alcohol molecule. And so that helps make it to where it's more water soluble and your kidneys can flush it out of your system. The smooth ER also stores calcium ions, and we're going to find out later on that ions are very important for muscle activity and also nervous system activity and things of that nature as well. The rough ER is called rough because it has those ribosomes that are stuck to it or bound to it. These ribosomes secrete something called glycoproteins. Remember, the ribosomes are the protein factories of the cell, and these ribosomes create these proteins that are then bonded to carbohydrates, and they create these fairly large molecules called glycoproteins. They also distribute transport vesicles, so this is where the ER kind of buds off or pinches off little portions of itself to make these little transport pods called vesicles. And inside those vesicles there could be proteins or other materials that are being transported from one part of the cell to another. And this is also the membrane factory for the cell. So the rough ER creates more of itself and more membrane material that's used for other purposes throughout the cell. Next on our list is the Golgi apparatus, sometimes called the Golgi body. And this is kind of like the shipping and receiving center of the cell. So again, if we're comparing the cell to a city, you might call this like the UPS or the FedEx of the cell. It packs and ships various materials throughout the cell. It's made up of various layers of flattened membrane. These sacs are called cisternae or cisternae, and it has a variety of functions. One of the things that it does is it takes material that the endoplasmic reticulum initially created, and it modifies it, it packages it, and it sends it out to various parts of the cell. It creates, again, some more of these large macromolecules, and then it also sorts and transports material from one portion of the cell to another portion, or sometimes outside of the cell to another part of the body. So here we have a model of the Golgi apparatus or Golgi body on the left, and then we have a microscope picture, a TEM of the Golgi apparatus on the right. You can see the various folds here, the layers that make up this membrane. It is kind of like a shipping and receiving center. It even has kind of a directional face to it, so that you have one side that's considered the cis face and one side that's considered the trans. Remember, cis means the same or same side. Trans means opposite or away. The cis side would be kind of like the receiving side, where material comes from the endoplasmic reticulum and it's received by the Golgi apparatus, and then it's processed in various ways throughout the Golgi apparatus. And then on the trans side, this is where new vesicles are broken off and that material is transported out. So this would be like the receiving side, and this would be like the shipping out side after the various modifications have been made. And it probably makes sense to say that the cis side of the Golgi apparatus tends to face the endoplasmic reticulum because that's the receiving side. So the ER might be, say, over here in this area. That would be connected to the nucleus that maybe perhaps would be up in this upper left corner. So you might have the nucleus in the upper left corner, and then you would have the ER, and then you would have the Golgi apparatus with the cis side facing the Golgi apparatus. I'm sorry, the endoplasmic reticulum. The third membrane organelle we're going to take a look at is called the lysosomes. And again, going back to our analogy, your lysosomes would kind of be like your recycle men or your trash men or your solid waste disposal units of the cell. The lysosome is a membrane sac that contains hydrolytic enzymes. And these hydrolytic enzymes can break down and digest various large molecules. So inside the lysosome are some pretty strong enzymes that can break down and hydrolyze proteins and fats, polysaccharides, and other materials. And the lysosome's job is to recycle or renew material. So let's say you have a cell part that wears out and breaks down. Well, the lysosome is used to basically tear that cell part down into its basic ingredients, so then it can be reused and rebuilt into something new or something different. Inside the lysosome, it has a pretty acidic environment, and those enzymes work best inside that acidic environment inside the lysosome. If you break a lysosome open, you're releasing those enzymes into the cell, and the cell material itself, the cytosol, has a neutral pH, and so those enzymes will not work as well inside the cytosol as they do within the lysosome. But if you were to break open too many of these lysosomes, you would still have a problem because you're letting out all these enzymes, and basically what would happen is the cell would be destroyed through self-digestion. So imagine that inside these lysosomes, it's like trash bags full of pretty toxic material, and you wouldn't want to break those trash bags open because you release too much of this toxic material, and the cell would basically digest itself, and that would obviously cause its death. Here's a quick little animation of a lysosome formation. So if you right-click and hit play, you should be able to watch that, and you should be able to do that in either the PDF or the PowerPoint presentation. Continuing on talking about lysosomes, there are some types of cells or single-cell creatures like amoebas and protists, and even some cells within our own body, certain types of white blood cells called macrophages, that actually eat by engulfing another cell. So they will kind of surround and engulf another cell, and this is called phagocytosis. And basically what happens is they surround this other cell and then form a food vacuole around that cell. So now they have engulfed that food source, and it's within their own cell body. And then a lysosome fuses with that food vacuole and releases enzymes that digest that molecule. So in the case of an amoeba, that might be how it eats. It surrounds and engulfs another cell or another microorganism, and then its lysosomes connect with that food vacuole and open and release enzymes that digest that material. With a white blood cell, it might, say, engulf a bacteria that has invaded your body, and then it will release the lysosome enzymes within that vacuole to destroy that bacteria. Lysosomes can also be used to recycle parts, like I mentioned. So let's say you have a mitochondria that wears out through continual use. Parts wear out in the cell in biology just like they do in mechanical systems. So let's say you have a mitochondria that wears out. Well, you may have a lysosome that surrounds that mitochondria. There may be a vacuole surrounding that mitochondria, and the lysosome fuses with that vacuole, releases enzymes that break that organelle down and get it back to its original constituent components, and then those components can be reused again by the cell. This is called autophagy. So it's like eating oneself, but you're basically breaking down and recycling parts of the cell. Here's a diagram of phagocytosis where you have a picture at the top, nucleus of the cell up here, and this little round object is the lysosome. So let's say, for example, you have a food vacuole where material is engulfed, and then the cell creates a vesicle, a vacuole around that. Here you have a lysosome that's filled with digestive enzymes, and it comes and attaches to this food vacuole, releases its enzymes, and then you have the breakdown of that large molecule into smaller portions that the cell can use. Here you have an example of the autophagy I was talking about. This is basically recycling of cell parts. Here you have a broken down mitochondria and a broken down peroxisome fragment, and see how they have been enclosed inside this vesicle. Then you can have a lysosome come to this vesicle and connect with it, release the enzymes, break that old mitochondria and peroxisome down, and then these building blocks can be reused by the cell to perhaps build a new mitochondria or to build a new peroxisome. The last major membrane organelle we're going to look at is called a vacuole, and vacuoles are kind of like storage containers or warehouses. So if we're comparing the cell to a city, this would be like your warehouse or your railroad storage car, something like that. They can be used for a wide variety of purposes. Most plant cells and fungal cells have one or more large central vacuoles, and animal cells will have them too, but they're usually not as large as some of the plant cell vacuoles. They are derived from the endoplasmic reticulum and the Golgi apparatus, so they are basically one of these vesicles that is pinched off from the ER, the Golgi apparatus, but they tend to be larger in size and they have a variety of uses. One of those we already mentioned. We talked about food vacuoles. So like when an amoeba engulfs another creature, it forms a food vacuole around that creature that it engulfed. So food vacuoles are formed from phagocytosis. Then we have what's called contractile vacuoles. Contractile vacuoles, you're going to see a video of in just a second. Lots of little freshwater protists, like this little paramecium down here in the lower right. They have contractile vacuoles. Man, that is like a tongue twister. Contractile vacuoles. Say that 5 times fast. And the job of these vacuoles is kind of like a pump. It helps pump water in and out of the cell to keep the balance of water and ions correct. And then finally in plants, you're probably familiar with the central vacuole in plants. Primarily it holds water and organic compounds and nutrients, and it also allows for the cell to be nice and rigid. It's partially the central vacuole and the cell wall is what allows a plant to stand upright without a skeletal system. It's what allows a dandelion stem to grow tall and fight gravity. So that central vacuole tends to hold quite a bit of water. You'll notice if you don't water your plants for a long time, they start to wilt and they limp and they go limp and they start to wilt to one side. That's because the central vacuole loses water and the cell becomes less rigid and starts to collapse. Here's a cool little video of a paramecium, and you'll see some vacuoles, some contractile vacuoles within the paramecium. So you're going to look for a couple of little dots here that seem to like open and close, and that's kind of like the paramecium is using that vacuole to either take in water or to get rid of water to control the amount within its body. Here you have a good picture of a central vacuole within a plant cell. A model to the left and an actual microscope picture to the right. You can see that central vacuole in a plant cell tends to take up a huge portion of the cell. Sometimes all the other organelles are kind of squeezed out to the side. Here you have the nucleus kind of being squeezed out to the side, and then you have, I see, 3 chloroplasts, and then the cytosol, the liquid material of the cell, is just all around the edges here, with the central vacuole taking up the majority of the space. On page 108 of our text is a great review illustration of all these various membranes and organelles, the endomembrane system. So the endomembrane system is made up of a number of different organelles, fairly complex system, tends to be a pretty important part of the cell, and it helps organize the cell into different parts and compartments. So here we have just a kind of a quick overview. We have the nucleus would be up here at the top. It is bound by what we call the nuclear envelope. That nuclear envelope is connected to the ER. Rough ER has the ribosomes. The smooth ER is without the ribosomes. On the very outside of the cell, you can see the plasma membrane out here. Add a few more things. Here we have material that's being processed in the endoplasmic reticulum. We have vesicles being broken off. Those vesicles oftentimes carry material to the Golgi apparatus. Remember, it kind of does the processing and packing and shipping out of these materials. Here's the cis side of the Golgi apparatus receiving that material. It gets processed, and then here you have the trans side, the shipping side, and it is squeezing off more of these vesicles, and it's transporting material maybe to another part of the cell or perhaps back to the ER or to somewhere else. It also could be transporting it or helping to create other vesicles like lysosomes and vacuoles. Here we have one of these vesicles that is actually merging with the plasma membrane, and that's a way that the cell can create materials and then move that material out of the cell, perhaps to go to another cell or into the bloodstream or something to that effect. The next concept is concept 6.5, and we're going to look at 2 very interesting organelles. These are oftentimes kind of considered unique, or sometimes many people think of these as their favorite organelles because they have to do with energy creation and energy change. So these are the mitochondria and the chloroplasts. The mitochondria and the chloroplasts change energy from one form to another. So both plant and animal cells have mitochondria, but only plant cells have chloroplasts. So you can find mitochondria in an oak tree cell or a dog cell, but only the chloroplasts would be found. They would only be found in the oak tree, not in the dog. Mitochondria are where you have cellular respiration, which is the breakdown of materials that uses oxygen and produces ATP, which is the basically the chemical currency, the energy currency of the cell. So the reason you have to breathe, the reason you have to take in oxygen, is because your cells have to do respiration, which is the breakdown of materials to release energy, and that requires oxygen. That takes place in the mitochondria. So Plants also go through cellular respiration. So even though plants in photosynthesis take in carbon dioxide and give up oxygen, they also do respiration, which is taking in oxygen and then breaking down materials to release energy. Chloroplasts, on the other hand, are found only in plants and algae and green living creatures. They are the sites of photosynthesis. You're probably familiar with that. And then we're going to talk about another kind of odd or interesting organelle called a peroxisome, and we'll find out what those are as well. The reason we talk about the mitochondria and the chloroplast together, not only are they both related to energy and changes in energy, but they also have some possible similarities as far as their evolutionary origin. For a long time, it wasn't clearly understood how mitochondria and chloroplasts came to be within the eukaryotic cell. And the reason is because they're somewhat odd, they're somewhat unique. Mitochondria and chloroplasts have some similarities with, say, bacteria or other single-celled creatures that live apart from cells or their own cell, their own living creature. The mitochondria and the chloroplast have their own double membrane, which is unique or interesting. They have their own ribosomes. They even have some of their own DNA. And that DNA within the mitochondria and chloroplast directs some protein production. So you have an organelle that's very much like its own living creature. They grow and reproduce somewhat independently in the cells, and the DNA material inside of them is separate and apart from the DNA inside the nucleus. So it's almost like you have another living creature, another living cell within your eukaryotic cell. Currently, the most widely accepted theory about this, and which we're going to discuss further in Chapter 25, is called the endosymbiont theory, And this is, at this point in time, the most widely accepted idea about how this occurred, although there's still, of course, science is ever evolving and ever changing, but right now this seems to be where the evidence is leading. So the thought is that an early ancestor of eukaryotic cells engulfed a non-photosynthetic prokaryotic cell, and instead of eating it, destroying it, actually developed this endosymbiont relationship with that cell that it engulfed. So basically imagine that an amoeba comes upon another kind of cell and engulfs it as if it was going to eat it, but instead of eating it, for some reason, somehow, it doesn't destroy it, it allows it to live inside of it, and they develop this symbiotic relationship that helps each other out. Well, if that were to occur as time went by, it might evolve into what we call a eukaryotic cell today, and that would be the mitochondria. So perhaps the mitochondria at one time was its own separate cell, its own separate creature that was engulfed by what became, through time, a eukaryotic cell. And then the thought is, well, if that occurred once, perhaps it occurred twice. So perhaps you have this new or early ancestor of a eukaryotic cell. It now contains what we would eventually call mitochondria. Maybe that happens again, and perhaps it engulfs another cell or another living creature that was photosynthetic, that did do photosynthesis and produce its own food. And again, instead of destroying and basically eating it, it allows it to live and develop inside. Well, if that's the case, that could become the chloroplast. That seems pretty far-fetched, but a lot of the evidence kind of points in that direction. So the idea would be that the mitochondria was at one time its own living cell, and the chloroplast at one time was its own living cell, and that both of these were taken in at various times in the evolutionary history of the eukaryotic cell, and they became organelles of eukaryotic cells. So here we have perhaps like a model. Here you have an early ancestor of a eukaryotic cell. You'll notice it has a nucleus, it has endoplasmic reticulum, it has some of the other parts, but notice it doesn't have any mitochondria, and if it was to eventually become a plant cell, it has no chloroplast. Well, perhaps these mitochondria were engulfed and became part of the cell. and then perhaps at some other point this type of cell engulfed a photosynthetic prokaryote, and it became a chloroplast. So if that's the case, you would have this, which would become an animal cell, and this would become a plant cell. So this is not exactly completely settled science. There's still some question as to how this happened or exactly when it occurred. But again, a lot of the evidence that we have now tends to point toward this theory. So let's take a look at these two interesting organelles. We'll start with the mitochondria. The mitochondria is found in nearly all eukaryotic cells, including both plant and animal. They have a smooth outer membrane, and then their inner membrane has a huge number of folds and turns and twists. The inner membrane is folded into what's called the cristae or cristae. The inner membrane has basically two parts to it because of all these folds. You have inner membrane space and something called the mitochondrial matrix. And inside all these folds in the cristae or cristae, that's where you have all these enzymatic actions occur that produce these ATP molecules, adenosine triphosphate molecules. And remember, the ATP molecules are the ones that are used for energy for the cell. Here we have a nice model and a nice microscope picture of the mitochondria. It does kind of look like a bean. It's got that smooth outer membrane, and then inside it's got all these twists and turns. This inner membrane is quite a bit of folded material inside. That's called the cristae or cristae, and you can see then inside it's got all these twists and turns, and that's where the ATP is created, where the ATP is made, where cellular respiration takes place. Here is a microscope picture, and again you can see all the inner folds inside. Here on the right-hand side, we have a microscope picture using a light microscope and some techniques to highlight some of the material of a single-celled protist. So this is a single-celled living organism. It does have a nucleus. You can see its nucleus right there, and it's got DNA inside the nucleus. But also you can see the little areas here, the mitochondria, and inside these dots are the mitochondrial DNA. So it has DNA that's separate from the nucleus, and that would seem to indicate that the mitochondria perhaps at one point was its own cell all by itself. Hopping over to the plant cells, we have a similar organelle as far as evolutionary background perhaps in the chloroplasts. And the chloroplasts, of course, are what contain the green pigment known as chlorophyll. It also contains a few other enzymes and molecules that allow photosynthesis to occur. So when you were a little kid and you're out playing touch football or keep away in your church clothes and you fall down on the grass and you get that huge grass stain on your pants, mom gets so mad at you. Well, what you've done is when you scraped across that grass, you crushed a lot of the cells in the plant, and that chlorophyll leaked out onto your pants and stained your pants. And that chlorophyll was located in a cell organelle called the chloroplast. The chloroplasts are found in the leaves and other green portions of the plant, and also in algae. So in a green stem of a plant, or obviously in the green leaves of the plant, or in the green algae. In the bottom right, beautiful picture here taken under a light microscope of plant cells. You can see the plant cell walls. They make this nice, in this case, sort of a diamond brick sort of shape. And then you can see the chloroplasts inside, kind of round discs containing chlorophyll. The chloroplasts have a couple different structures inside, and we'll look at a kind of a zoomed in model in just a second. They have these sacs called thylakoid membranes. They are in stacks. So if you can imagine round sacs stacked in piles, these stacks are called granum, and then they have a fluid around them called stroma. So inside the chloroplast you have thylakoid membranes, which are stacked together to form granum, and then you have stroma, which is the fluid inside. The chloroplast is one of a group of organelles called plastids. Some plastids are used for storage, like, for example, in the potato. If you have a potato plant, the potato itself contains plastids that are used to store starch. Some plastids are used to store different types of pigments, which might give the flower or the fruit its color. So there's different types of plastids within the plant cell, and it's different various organelles that are kind of related as far as their structure. And one of these is the chloroplast. Probably the most important one, of course, because the chloroplast is the one that allows the plant to capture the sun's energy and create its own food source. Here's a great model showing the chloroplast. So you have this entire structure would be the chloroplast. Inside you have these thylakoid membranes that are stacked up on top of each other. One of these stacks is called the granum, and then the fluid around the granum is called the stroma. And then you also see some DNA and ribosomes inside there. So again, this would seem to indicate, because it has its own DNA, its own ribosomes, it has a double membrane, that seems to be very indicative of the fact that perhaps this chloroplast was at one time its own separate photosynthetic cell, its own separate photosynthetic living creature all by itself. And then here we have a TEM of the chloroplast. Kind of shows a cross section, some of the same things. Here we have same pictures, but on the right hand side we have an algae cell, and the chloroplasts are showing up in the algae cell. They've been marked with this red dye, or they've been perhaps color-coded with a computer to make them show up. So far we've talked about the mitochondria and the chloroplasts. We kind of group those two together because they both have something to do with energy, and also because perhaps they both came to be within the eukaryotic cell in the same way. Perhaps early in the history of the evolution of the eukaryotic cell, they were engulfed and they developed this endosymbiotic relationship with the cell. So that brings us to the last one we're going to look at for right now, called the peroxisome. Peroxisomes are very specialized little metabolic compartments. They are within a single membrane, not a double membrane, within a single membrane, and they produce hydrogen peroxide, which is actually a fairly dangerous substance, especially within inside the cell. They produce hydrogen peroxide, and then they also have the ability to convert that hydrogen peroxide to water. They have a wide variety of functions, and it's not known exactly how they relate to the other organelles. Peroxisomes are still a bit of a mystery. Some scientists think that they also were engulfed by the early cell and became an organelle, and that at one time they were separate individual cells by themselves. But other scientists disagree with that. So peroxisomes are a bit of a mystery. There's still some scientific argument going on. And this is a good example of how science proceeds. So no, we don't have the full picture yet, but as we gather data, we'll basically decide one way or the other what we think, perhaps is the most well understood idea, which one has the most facts to back it up. So this is kind of a hallmark of science and the scientific method, where we proceed as best we can with the data that we have, and as we gather new data, we change our thinking. And a lot of people tend to think that that's a weakness of science, that it's kind of wishy-washy and changes, but that's completely the opposite is true. That's actually one of the strengths of science, that you are willing to change your thinking and change your ideas based on evidence. And as new evidence presents itself, you conform to that new evidence. So peroxisomes are unique organelles. Not exactly sure how they fit in quite yet, not exactly sure how they came to be within the cell, but there's still some scientific debate going on about exactly how they came to be there. Last slide for this portion, just showing a TEM microscope view of peroxisome. So here you have, this is a plant cell. We know that because we have chloroplasts here. Here's a mitochondria, and this is actually the peroxisome here in the middle between the two. Again, the peroxisome gets its name because it has the ability to produce hydrogen peroxide, which again is a heavy oxidizer, which can be quite damaging to the other cell parts. So it's separated from the rest of the cell by a single membrane. And the debate is, did this peroxisome become part of the cell in the same way that the chloroplasts and the mitochondria did, or was it always part of the early eukaryotic cell? And we're not quite sure yet, but as we gain more information, we'll clarify that. We're going to stop right here, and then we're going to move on to the framework of the cell, kind of the cytoskeleton, and that will finish off Chapter 6. Be back in a sec.